Uh, Everybody, thank you so much for joining us today at Hatfield's Research Seminar. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon, and I will be your host for today. Just want to let everybody know that this is a Zoom platform, but we have turned off and disabled your cameras, mics, and screen shares for those folks that have limited bandwidth so they can participate with us. That does mean that we hope that throughout this talk that you communicate with us using the chat function. So you can find that chat function either at the bottom or the top of your screen, and you can put in your questions there, and today's speaker will answer them whenever you put them in. So please encourage you to ask questions whenever they come to you. I wanted to do also let you know that we're recording today's seminar. So if you missed something, you want to see it again, or if you'd like to share it, it'll go up on Hatfield's past seminar page in a few days. And I just put that link into the chat so you can see that and also see where the chat happens to sit when you open it up on your screen, if you would like. Wanted to do a quick little promo for next week. We have same time, same place next week. We have a seminar speaker, Brian Fulfrost, who will be talking about mapping marshes and mudflats from space. So interested in those kind of things, come join us again at 3:30 next Thursday. I also wanted to let folks know that we have our monthly Science on Tap next Thursday. That starts at 6 p.m. And Erica Flashman is the director of the Oregon Climate Change Research Institute at Oregon State University, and is going to be talking to us about last summer's wildfires and the science and societal response to changing dynamics of wildfires on the coast. So that should be a really interesting talk and a very timely talk as well. So if you're interested in any of Hatfield's events, you can Google or use whatever platform you use to find HMSC's home page. Go to the bottom and you'll see a calendar of events with all the login information that you need. I also wanted to just let folks know that we are going to continue the virtual seminar series throughout the summer, and I'm booking speakers now. So if you would like to volunteer any of your colleagues, let me know and I will reach out to them and see if they're interested in giving a talk. We have a few more students that are around in the summer, so if they're interested in connecting with anybody at Hatfield or with any of our students, please let me know and I will get them on the schedule. But for today, while we're all here, we have Beth Phillips from NOAA's Northwest Fisheries Science Center, who will be speaking about using archival data. But first, a little background about Beth. She completed her PhD at the University of Washington School of Aquatic and Fishery Science in 2018. She has a master's from Moss Landing Marine Laboratories in San Francisco State University and a bachelor's in biology from Western Washington University. Go Vikings. That is also where I have my bachelor's from. Prior to joining the Northwest Fisheries Science Center as a research fisheries biologist, she was also a National Research Council postdoc. Her current research is focused on using fisheries acoustics to study the California current. And I think she'll be talking about that a little bit with us today. So Beth, the floor is yours. I hand it off to you and take it away.
Okay, thank you, Cinnamon. Can everyone hear me? I'm assuming so.
You sound great.
Okay, and yes, I wanted to reiterate that if you do have questions, I'm happy to stop and pause and answer them throughout my talk. I'll try to keep it a little bit more informal that way. So thank you, Cinnamon, for your nice introduction. I'm excited to be here to present with all of you on the research that I've been doing with NOAA Fisheries. Before I get started, I did want to pause and acknowledge that I am speaking with you from the unceded ancestral lands of the Duwamish people, the first people of Seattle. And I honor with gratitude the land itself and the Duwamish Salish tribes. So data sets that span broad temporal and spatial scales can provide really important insights on ecosystem variation, particularly in light of climate change, and also inform ecosystem-based fisheries management. As Cinnamon mentioned, I am a research biologist at the Northwest Fisheries Science Center, and I work with the Fisheries, Engineering, and Acoustics Technology team. And we are focused on doing research in the California current. So that's the work I'm going to share with you today. So the California current system is a productive eastern boundary current that forms when the North Pacific current bifurcates near Vancouver Island and flows south all the way towards Baja California in Mexico. And this is a figure showing the California current's flow from northern British Columbia down along the west coast. The California current is dominated by coastal wind-driven upwelling, particularly in the spring, which results in very high levels of primary productivity. That said, the California current is quite complex. There is a lot of different coastal jets and eddies that form as it flows south, and there's a lot of large volumes of fresh water inflow, particularly from the Columbia River, that can also make the physical oceanography quite dynamic and quite complex, and that can influence the biological community as well. The high levels of primary productivity in the California current support a wide and diverse food web, as shown here on the left. And this includes commercially important fish species such as Pacific salmon, many species of rockfish, northern anchovy, and Pacific sardines, market squid, and of course, Pacific hake. In addition to supporting all of these commercially fished species, the California current also supports a number of a diverse assemblage of marine mammals, including humpback whales, blue whales, as well as seabirds such as this Cassin's auklet shown in the lower right of the food web, as well as common murres and sooty shearwaters and many other seabird species, as well as sea turtles. What you'll notice when you're looking at this food web is that right smack in the middle is krill, or large zooplankton, euphausids, also known as krill. And all of these species that are shown in this food web feed on krill, and they are a quite important part of the California current ecosystem, and it's been the focus of a lot of my research. So as I mentioned, I work with the FEAT team, and one of our primary charges is to conduct biennial surveys of Pacific hake in support of stock assessment and fisheries management of that species. Pacific hake are the largest by volume fishery on the West Coast, and they're commercially fished in both US and Canada. And as such, we operate joint surveys that we coordinate with Fisheries and Oceans Canada to obtain accurate estimates of hake biomass for the stock assessors. And the hake fishery is managed jointly between Canada and the United States. Pacific hake are thought to spawn off Southern California, as shown in the left-hand figure there. The gray area off Southern California is their spawning grounds during winter. And they're thought to migrate north along the west coast during spring and summer. And those are the arrows now showing them moving further north. And depending on ocean conditions, as well as the age of the fish, some of these animals migrate further north than others. It's thought that the larger adults migrate further north into northern Washington and British Columbia. And also during warmer years, it's been shown that a lot of the hake will move further north compared with cool years. So the goal of our survey is to estimate biomass of the entire population. And so we have in the middle map here is now showing our survey area. And you can see that our line transects begin in Southern California near Point Conception, and we work our way all the way north up into British Columbia and even past Haida Gwaii. The northernmost extent of our survey is determined by the northernmost extent of the hake themselves, which we're detecting on our surveys. We also, the westernmost extent of our transects are also determined by the extent of hake. So we're sort of focused on trying to detect and estimate biomass of the entire population. Another nice feature of this survey is that we operate our transects running east-west, and we cross continental shelf, shelf break, slope, and even offshore habitats. So we're sampling quite a large area of the California current. So to quantify hake biomass, we use active fisheries acoustics or scientific fish finders to detect hake aggregations. And we're typically operating these echosounders at multiple frequencies, including 18, 38, 70, and 120 and 200 kilohertz. And I'll touch on why that's important in a little while. I also wanted to note that these surveys have been operating jointly in their current form and design since 2003, and we're actually getting ready to head out on our 2021 survey in about a month or so. So this is a depiction of the NOAA research vessel that we work on, the Bell M. Shimada. And this is the portion, the vessel we use on the US portion of the survey, I should say. So when Hake aggregations are detected, we sample the fish with a midwater trawl, which allows us to verify the species composition as well as age composition, their sex. We can collect tissue samples for genetics as well as stomach content data. And you can see the beam of sound here coming from the hull of the Shimada is illuminating 3 Hake there, and then the midwater trawl. I just wanted to note, as I'm sure you all are aware, that this is not to scale. We've also recently integrated sail drone capabilities into our survey design. And this allows us to take advantage of new technologies to survey areas beyond where the Shimada can go. For example, further offshore, where there may be some Hake occurring that we're not detecting. And it's really a nice way to complement our ship-based survey by using these drones. And that's been going since 2018. And I wanted to point out that because our survey covers nearly the entire California current system, we have the opportunity to collect data on many other aspects of the ecosystem. And that allows us to contribute to a much better understanding of the state of the ecosystem and, of course, make forecasts and predictions of how that might change in the future. So, for example, now I'm just showing a CTD rosette coming off of the Shimada. We are able to sample ocean conditions, including temperature, salinity, and dissolved oxygen through the water column. And we do this predetermined CTD sampling stations all along the coast. We also have an acoustic Doppler current profiler system that collects data on the strength and direction of different currents, which can be used to inform estimates of, for example, cross-shelf transport and distributions of larval fish and zooplankton. And as I mentioned earlier, we're collecting data at many different frequencies using our echo sounders. And this gives us the ability to see many different fish and zooplankton, in addition to the Hake, that's our main focus. We've been conducting research on detecting rockfish aggregations as well as mesopelagic species. And in particular, the focus of my work has been on looking at euphausids or krill and developing a time series of data. I also wanted to point out that all of this data, which amounts to hundreds and hundreds of gigabytes of data per survey, upwards of terabytes of data, is archived, and we store that. And it presents a really exciting sort of treasure trove that we can look back through and begin sort of resurrecting to start thinking about how we can address new questions that are emerging. So I just wanted to show you a depiction of what we are doing when we're out on the boat. So this is an echogram. So this is the acoustic sound being scattered back to the vessel. And this is sort of just depicting as the vessel moves along a transect. The black here is the sea floor, and it's moving from about 500 meters water depth on the left-hand side of the echogram and coming up to about 200 meters and then dropping down off the bank again as the vessel moves. And this is about 8.5 kilometers length of transect. This is what we would see on the 38 kilohertz echo sounder. And the red and orange colors there are basically just showing things that are scattering sound much more strongly. In this case, it would be fish that have swim bladders. And what we do is we then put a net in the water and we sample that aggregation that we're detecting. And this is about 300 to 400 meters water depth to verify that it is indeed Hake. And in this case, that is indeed what it was. But what's really been neat for me is to go back through this data and start looking at the other acoustic frequencies that we have. And this is showing the exact same slice of transect, but at a different frequency, in this case, the 120 kilohertz frequency. What you can see is that as the vessel's moving along, There's another nice big band of strong scattering, again, the red colors, between 100 and 200 meters water depth. And that actually, as we know, because zooplankton scatter sound much more strongly at higher frequencies, we were able to verify that that is a nice big layer of krill that occurs right above these aggregations of hake. So that was sort of what my postdoc, what I came on to work with NOAA Fisheries to do, was begin reprocessing this treasure trove of acoustic data. My goals were to develop methods to classify krill in this archived time series going back through to 2003. I wanted to work out if there were ways to efficiently process hundreds and hundreds of gigabytes of this data, and then finally quantify the spatiotemporal variability in krill abundance and distribution along the coast. So one of the things, the first thing I needed to do was figure out how to identify krill. And I turned to some methods that were developed by other scientists in Alaska, and I've also been working with scientists at DFO to basically use what's a method called frequency differencing, where you can basically subtract the amount of sound scattered at different frequencies to remove a lot of the noise from our echograms and really just zero in on our species of interest. In this case, it was euphausids. And so I was focused on looking at the 120 kilohertz echo sounder data and pulling out what we could verify were indeed krill. And so you notice in this echogram now, quite a lot of the colors disappeared, a lot of that noise went away. And that was really important to make sure that what we were quantifying was truly could be classified as krill and not anything else as far as smaller fish species or other types of scattering organisms. So I spent a lot of time, speaking of diving through treasure troves of data, that took me about a year to go back through that time series. And I mentioned that this survey started in 2003. Unfortunately, we weren't able to use data from 2003 or 2005 for the full extent of our analysis. So most of the data you'll see from here on out is from 2007 through 2019. And I can certainly answer questions about that later if you want to talk about that. I'm also showing you sort of the area I was focused on for quantifying krill, which was just from 50 meters water depth down to 300 meters water depth. We excluded data that was deeper than that because the signal to noise ratio at the higher frequencies, it gets too hard to discern the noise from any of the signals. And similarly for 50 meters, we knew that at least for the euphausids that we were focused on studying, they typically occur during the daytime, deeper in the water column. I also wanted to mention that the two species that we developed these methods for are the two most common species in the California current, meaning Euphausia pacifica and Thysanowessa spinifera. So what did I find? I thought I would just jump right into some of my results. So one of the nice things about acoustic data is that we have really high resolution information. You know, we are pinging our echo sounders at least every second, if not faster. And it's picking up scattered sound all the way through the water column. And so we can start thinking about how to bin the data in different ways, sort of horizontally along transects and then vertically. And this is just a figure now showing the distribution of krill through the water column for all of the different years that I was able to analyze. And so you can see the depth here going from 50 meters down to 300 meters. And what I wanted to point out here is that in all of the years that we have, first of all, the distribution of krill is quite uniform. If you look, there's really not any extra peaks down at 300 meters, which I was a little bit concerned about if we were missing anything. The other thing is that there was really no difference in the average depth of krill through all of the years. Their average depth is about 150 meters, and there was no significant difference in that. Even in 2015, which I had hypothesized they may have gone deeper, because we know that, during the marine heat wave of 2014 through 2015, we know that a lot of critters were pushed further down in the water column. We didn't see that for the euphausids. But what we did see now in this figure, you can see the spatial distribution of euphausids along the coast. And the pink dots here are just representing the relative density measured as nautical area scattering coefficient, or NASC, if you've heard of that before, referred to as for fisheries acoustics. It's sort of the relative density of krill. The larger the dots, the more krill were in that location. And you can see that there was actually quite a bit of spatial variability. The black dots on each of these maps just represent sort of the geographic center of gravity. If you just sort of took the average location for the whole coast, that would be where they would be located. And that's sort of typically off of Washington or Oregon, with the exception of 2015. What you can see here is there was really not a lot of krill at all along the coast of Washington, Oregon, or California. However, there was actually relatively high numbers of krill detected in the northern part of the California current in British Columbia. And you can see that the shift in the distribution, the center of gravity, also moved further north because most of the krill were found up there.
Beth, we have a question in the chat. Can you talk a little bit more
Sure.
about when and how these surveys were conducted? Is it day and night or both to account for vertical migration?
Yes, thank you for that question. So these are all daytime surveys. And I should say that the surveys typically start, as I mentioned, in June. And we work our way up the coast throughout the summer, ending in British Columbia and typically in like mid-September. And all of the data that I've analyzed is only for daytime observations. So yeah, we don't have any information right now, at least about diurnal vertical migration or worries about anything being up near the surface if we were out there at night. So this is all daytime distributions. Any other questions?
No, that was it for now. Thank you.
Okay. So this is now just showing sort of condensed through space now, just the overall distribution of, or density, excuse me, of krill through time, through the time series of data that we have. And you can see that, again, for 2015, it really stands out. There was just very low numbers of krill along most of the coast, particularly the US West Coast. What I also wanted to point out, though, is that you can see 2013 was quite high, the abundance was quite high, but also 2017 and 2019 have sort of just average amounts of krill. You would have expected, given what we know with more recent marine heat waves since 2015, maybe lower numbers of krill, but it seems as though for some reason, we're not quite sure yet why, but it seems that the krill, at least populations in those 2 years, weren't as affected as they were in 2015. So I also wanted to start thinking about how to condense all of this information. And one of the ways I did that was using kernel density estimation to examine basically hotspots of krill through the time series. And so what I'm showing you here is basically a surface of krill sort of along throughout the entire time series. And the yellow is the 95th percent quantiles. And those are identified as basically hotspots. And what you can see in this figure is that there's a quite large hotspot that consistently occurs off of the Strait of Juan de Fuca and the border between US and Canada. You can also see some hotspots off of the Oregon coast near the 200-meter isobath, as well as near Cape Mendocino in Northern California. But you also see, if you look closely way up north, there's a hotspot of krill even all the way up there into Haida Gwaii. So krill basically are present throughout the California current. It's been really interesting to start looking at data with the joint survey going into Canada and looking at the distribution all the way beyond sort of the border with Washington and Canada, and looking up there and seeing that there's actually quite a good population of krill up there. Oh, and here I labeled the two for you to see where Cape Mendocino is. So I wanted to step back and start asking the question after I sort of processed all that data for my postdoc. You know, I mentioned before, we're seeing these krill layers at the exact same time we're seeing a lot of hake aggregation. So what does that mean for these predator-prey interactions? And in particular, what does it mean for this commercially important species, Pacific hake? So I have now going to sort of switch gears and focus on looking at distributions of krill in relation to Pacific hake and sort of comparing, because we are collecting this data literally at the exact same time. We have this great opportunity to start comparing direct predator-prey potential interactions. So here again is the plot showing krill distribution on the left, and I've stretched out the y-axis down to 500 meters now, just so that it would match the hake distribution, which is on the right in the gray bars. And what you can see is that the mean depth of hake was much more variable than krill. They kind of move up and down in the water column during different years, and about 100 meters deeper than krill on average. And so this is just another way to show it a little bit more condensed. So we're not looking at all the different years now, but you can see really a nice distribution of krill overlaid with hake. And you can see sort of the center distribution of krill is about 100, 150 meters water depth compared to the hake. And as I mentioned, you can now see that hake and krill, their depth distributions were not correlated at all. And this is primarily driven by the fact that krill really aren't moving very much compared to hake, which are moving down deeper into the water column for certain years. By, the range is about 100 meters or so. So then I did the same thing, looking at hotspots, comparing hotspots of krill to hotspots of hake. And now you can see the map on the right showing the hake. And what you see here is that they also have some hotspots, but some of them are in similar spots where we're detecting nice big aggregations of krill, like, for example, off the Oregon coast, where many of you are probably located. There's also some hotspots, though, down in Central California off of Monterey Bay and San Francisco Bay, Cape Mendocino. But the really interesting part for me was that there was really not any hake hotspots found up in British Columbia at all. So although the krill are there, certainly not as many hake are there, at least when we were out there, when we're out there surveying during the summertime. So I wanted to look at direct sort of co-occurrence or spatial overlap, since we have this data collected at the exact same time. And so now I'm just showing you sort of maps of the co-occurrence of hake and krill in the same areas. And in this case, it's 25 square kilometer cells that I mapped this out into. And what you can see here again is that it's actually quite variable. The average number of cells that had both hake and krill across all of the time series was about 35%. But in some years, for example, in 2007, the co-occurrence of hake and krill was quite high, upwards of 48%. But again, you can see in 2015, it really stands out that the co-occurrence of hake and krill is quite low, only about 17%. So this got me thinking about what physical conditions are influencing hake and krill in the California current. As I mentioned earlier, our surveys cross a wide range of bathymetric habitats, including shelf break, slope, and shallow basins that are on the continental shelf, as well as, of course, submarine canyons. And so I used a bathymetric map to basically extract information about both water depth, distance to the shelf break, distance to submarine canyons, et cetera, to sort of start thinking about maybe that's an important factor explaining the distribution of these two species. Of course, we also wanted to focus on some biological components, and so we also looked at chlorophyll A and concentrations of chlorophyll A along the coast while we're out there. So I used Aquamodus satellite data to match chlorophyll A concentrations to the locations where we were observing all of the hake and krill. And then finally, of course, with an eye towards the effects of the marine heat wave in 2015, but also thinking towards future changes to ocean temperatures, I wanted to look at temperature influences on hake and krill. And for this one, what I was able to do, because our survey collects all of this great oceanographic data in addition to our acoustic data, is I used temperature measured by our CTV cast at 100 meters water depth across all of the stations. And so we were able to generate basically a surface map of temperature at 100 meters. And from that, knowing, that what we know about how temperature varies across the California current, we took the anomalies of those 100-meter water temperatures to look at to control the spatial variation of that, but then look at the influence of temperature on both krill and hake. And so I'll show you some results. Oh, yeah.
We have a question that you said you would talk about it maybe a little bit later, but it's coming up. Can you talk about why there was no survey data being presented for 2014?
Yes. So our surveys are biennial. So we operate typically in odd years. So 2003, 5, 7 onwards. We did do an additional survey in 2012. But unfortunately, and then 2013, but unfortunately, our next survey wasn't until 2015. So we didn't get 2014 or 2016 data to sort of bookend that marine heat wave, unfortunately. And that's just the way our surveys are designed. We conduct our stock assessment surveys every other year. And then on the even years, we're out there, but we're conducting research surveys where we're not surveying in a standardized area as we do for our stock assessment. So yeah, unfortunately, we don't have that data for 2014. It would have been great, but we don't have that.
Thanks.
I think that answered the question.
Yeah, definitely. Thanks.
Okay, so I'm going to just show you some outputs now from generalized additive models that we looked at for just now the presence of krill and the presence of hake. And in this case, I'm showing the influence of bottom depth on their presence. What you can see is that for this variable, it's really similar. Both hake and krill, as we know, occur near the shelf break, around 200 meters water depth. And you can see that sort of peak in, around between 200, 400, and a little bit closer to 500 meters water depth on the probability of hake and krill being present. And that's shown in the blue line. And it was really similar as well for the distance to the shelf break. And so they're really sort of centered right around 0 here. It's shown on the x-axis here, sort of being 40 kilometers either offshore or onshore of the shelf break. And you can see that both of these species, the probability of presence is, sort of reaches a peak right around 0, so being right on top of the shelf break. That wasn't too surprising. I also wanted to show you the influence of chlorophyll. This is the log of chlorophyll now. And again, they're sort of especially for krill, sort of their probability of presence is sort of occurring at intermediate values of chlorophyll, not a really strong influence of low or high chlorophyll values. And again, this makes sense given what we know about upwelling strength, primary productivity along the coast, and sort of the combination of transport, strong upwelling winds, and transport of zooplankton. There might be a slight mismatch between where there's really high levels of chlorophyll A in primary production and where we're actually locating the krill. But what was really interesting is that we found this sort of spatially variable effect of temperature. And so what I'm showing you here on the left is a map of the effective temperature on krill. And you can see that the blue is basically showing that when there's lower than average temperatures at 100 meters, the probability of krill being present is greater. Again, we know that these two species of krill that we've been focused on tend to be more abundant when there's cooler waters. The opposite was observed, though, up near Hake, where when temperatures were warmer than average, there was increased probability of krill being present. And that's still something that I'm still pondering why that might be. Let's see. And then, something happened. Well, I wanted to show you, let's see if that's works. Oh, yeah, sorry, I forgot to mention the Hake component. So the interesting part is for Hake, you can see that it's a little bit different story where the effective temperature is sort of matches in California, in Southern California, that when it's cooler than average, the probability of Hake is greater. But it gets a little bit more mixed along the Oregon coast and British Columbia, where actually the probability of hake being present goes up when the temperatures are above average. And I think this speaks to a little bit to the, what I talked about earlier, the ability of hake to migrate further north when warmer ocean conditions occur. And then the opposite was observed in very far north for hake, where the temperatures are cooler than average and the probability of them being present there. So this sort of suggests that there might be something going on with temperature that might be influencing the probability of hake and krill occurring at the same place in the same time if temperatures were to change. And so that's now what we took our generalized additive model outputs, and then we just decided to make some predictions based on what we saw, controlling for all of the variables for bathymetry and chlorophyll A, and just looking at the influence now of temperature to make some predictions. And this is showing just sort of what we would expect under average ocean conditions, where temperatures were allowed to stay sort of at their long-term average for the time series that we have. And we're predicting about 22% co-occurrence. So in 22% of the area of our survey, we would expect to see hake and krill co-occurring. And you can see that a lot of the co-occurrence is predicted to be in Northern California, and again, Oregon near the continental shelf break. When we allowed the temperatures to drop by 1 degree below the long-term average, we saw that the probability of co-occurrence goes up on average to about 40%. And the probability of hake and krill co-occurring is quite high throughout most of California waters, and again, quite high off Oregon. Again, not much in Oregon and Washington, but again, you start seeing a little bit of a hint of something going on in the northern part of the survey. But then what was really interesting is when we had temperatures drop to 1 degree below the long-term average, and we made predictions, co-occurrence dropped to about 15%. And it was really concentrated near Cape Mendocino in Northern California, and then also off of Vancouver Island, which again is where we were observing some of the hot spots of hake and krill near Cape Mendocino, and again where we know the hake will tend to migrate when temperatures get warmer. So just to summarize now, as I've shown you, hake and krill tend to co-occur in about 35% of the area. But I just showed you that temperature can have a strong influence on that co-occurrence. And of course, this has really important implications for hake growth and recruitment to the fishery. And information about krill abundance and distribution has the potential to really be really important information for trying to implement ecosystem-based fisheries management for this species. And we're working on that right now with some work I'll touch on in a moment. This also indicates that assuming that hake are consuming krill for most of their diet, that the demand for prey during warm years might be much higher than in cool years, simply because their distributions are shifting and the abundance of krill is much lower in these warm years. So their demand for prey might be much greater, which might affect, of course, their growth, but it also may increase competition for alternative prey resources. So if there's prey switching to consume other species of zooplankton or fish, it's also probably going to increase competition with other predators as well. This is sort of ongoing work that we're thinking about now. One of the things I've been doing is resurrecting. So I've talked to you about how I've resurrected a lot of acoustic data, but I'm also now working to resurrect some of our biological data. I mentioned that we take samples of stomach contents during our surveys, and now we have a database of hake stomach contents that another member of the FEAT survey team, Alicia Billings, has worked very hard on putting together, and it eventually will go all the way back to 1998 and all the way through present, and it includes stomach contents from Canada, Alaska, and the United States. So it's a really exciting thing for me to be thinking about, the ability to ask new questions about how do hake respond to variation in krill, and I'm going to be able to look directly at their diet and look at the distribution of krill at the exact same time we were sampling the hake to see if they are indeed switching to different prey, they're consuming fewer krill overall, their growth is less. So these are some of the questions I'm starting to switch gears and start focusing on now. And of course, thinking about how this might all influence the recruitment dynamics of this important fisheries.
So Beth, there was a question that came in about your GAM functional response curves. And I missed it before you went to this slide. But what did the GAM functional response curves of krill and hake look like relative to temperature?
So the GAM, it was that two-dimensional surface that I showed was, so within our GAM, we included a latitude and longitude smoother in addition to temperature. So yeah, I don't have like a one-dimensional plot of those. We did do some models with just one dimension of temperature, excluding space, but knowing from some previous work that another postdoc had done about the influence of temperature, sort of the spatial influence of temperature, we wanted to include that sort of 2D spatial component. So, but I'm happy to show the other plots later if that helps.
Okay, thank you. And then we have a question about what are some of the alternative prey of hake, or is that a future seminar coming our way?
Yeah, so I'm so excited to dig into the diet data. We know in general that hake are pretty voracious predators. They have quite a large gape, and so they will essentially eat whatever they can get their mouths around, including other hake. That's actually an important component of their dynamics, is that they are cannibalistic. So yeah, alternative prey could mean anything from other coastal pelagic species like anchovies and herring are an important component of their diet. Maybe sardines, hake, certainly krill. Yeah, I think once I dig into the diets, I'll be able to tell you more, but I can tell you for certain right now, at least from the quick look I've taken, there's probably at least 60 different species of animals that they've documented to be consuming. Rockfish is another one. So, before I move on from this one, though, I just wanted to highlight, we do have a postdoc, if anyone in the audience is interested in looking at the influences krill and cannibalism and on hake recruitment dynamics. We have a postdoc through Seacoast, which is the new program at the University of Washington, the joint program, to develop some multi-species ecosystem models to start evaluating sort of the influence of all of these different components on their population dynamics. So get in touch with me if you're interested in that. Okay, and then, okay. So finally, I wanted to mention, that this work has been focused primarily on the influence of krill on hake, but stepping back to the rest of the food web, we know there's a lot of other predators in the system, including salmon, rockfish, porous humpback whales, blue whales, and we're not sure yet if these results will be comparable for other predators. It's certainly, now that I've developed this time series of krill, there's a lot of really exciting questions that we can start answering or start asking about, the influence on salmon, for example, or distribution of humpback whales when there are fewer anchovies, are there more krill, and looking at shifts in distribution related to what we have for information on krill. So I think there's a lot of exciting research that we can continue talking about. And then finally, I just wanted to touch on the fact that we are bringing in some new projects to our FEAT survey. So this summer, we're going to start collecting information on distribution of marine mammals and seabirds along the coast. I mentioned rockfish and mesopelagics. We're also going to be delving deeper into some of the archive data to start classifying and identifying those. And we're starting to do more seafloor habitat mapping. We have a number of collaborative research projects that are ongoing on the Shimada, and we're always excited to do more collaboration. And if you want to follow along, we have at sea blog called The Main Deck that you can Google. The Main Deck at NOAA and you can get updates on where we're at and what we're doing during our survey. So with that, I just wanted to conclude and sort of reiterate the fact that, there's this treasure trove of acoustic data and oceanographic and ecological data that has been really fun for me to sort of dive into and pull out really interesting data from. I wanted to point out that reprocessing this data is not necessarily an easy task. As I mentioned, it took me about a year to get a lot of these programs up and running and trying to sort through all the data. And but it can, regardless, it can be quite useful. And I think there's a lot of other uses for this archived data, especially as we start thinking towards climate change and new emerging questions with shifts in species distributions and fisheries management that needs to be adaptive to this new the new world we're facing. So with that, I wanted to just thank everyone for tuning in and acknowledge Fisheries and Oceans Canada, the collaborators there, as well as many different scientists at the Northwest Fisheries Science Center who've helped me over the last few years work through all of this, this research and this project. And of course, the NRC postdoc program, which got me going with this in the beginning. And with that, I'll take any more questions that might pop up. And again, thank you for tuning in.
Great, thank you so much, Beth, much appreciated. Folks, you're welcome to put in any questions into the chat and we can answer them now. And Beth, and this might be going a little bit too deep, but I was just curious, when you were doing your hotspot analysis, you were talking about doing it in an aggregate. I was wondering if you looked at it as a yearly series to see if there was any variation related to temperature that kind of validated some of the model work that you did later.
Yeah, so yes, I did. I didn't show the individual sort of hotspots for each year, but that's something I've been thinking about is looking at the locations of those hotspots in relation to specific things like submarine canyons, for example, or shifts in temperature. I have not yet done that analysis, but I think that's certainly something that I would like to do in the very near future.
Yeah, it seems like it could help validate some of the other aggregate work that you were doing as you were talking about temperature shifts.
Yeah.
All right. We're a little quiet in the chat, but feel free to put things in if you have them. Otherwise, I'm going to say, Beth, have a really amazing trip. I'm glad that you're able to go out this summer and keep the data set going. And we wish you luck with all of your work. And just make another plug if anybody out there is interested in postdoc work. This work seems like it's rich with lots and lots of things that can be explored and pulled apart. So just thank you very much. They're coming in so quickly now. Hang on, let me see if I can catch a couple of these before we lose them. We're getting questions. Walleye pollock extend as far south as central BC. Do you think pollock and hake might interact and not just compete? Yeah, that's a good point. So yes, there are walleye pollock that are detected even inside Puget Sound sometimes. Yeah, maybe hake are consuming young pollock, or vice versa, pollock are consuming large hake, if that's what you're meaning by interaction. Yeah, there might be some interactions there. I don't know. I don't think the abundance of pollock is very high relative to hake. And particularly, as we know, pollock are shifting further north. There may be fewer of them now than there have been in the past. But yeah, that is a good question. Yeah, and actually, I think the fishery might have some good information on that too. I don't know if they are, they're targeting hake, but if they ever catch pollock, there might be some interesting information there. Nice. So another question came in. You talked about co-sampling with a trawl net. Is anybody sampling the krill aggregates that you find directly?
Yeah, that's a great question. So I kind of brushed over the methods that I used to classify krill in the acoustic data, but DFO did do some targeted samples of krill with a paired ship with an echo sounder on it, where they, were detecting aggregations that they suspect were krill, and then they used net samples to collect krill and confirm the species, either Euphausia pacifica or Thysanowessus spinifera, estimate their length and then their target strength, and then sort of use that to develop the methods that we were able to use to classify krill. That said, those surveys that were really focused on developing the methods were conducted off Vancouver Island between 2014 and 2016, I think. Since then, we have done some sampling of krill aggregations, particularly on our 2017 survey, but it's not nearly as standardized as we are doing for the acoustic trawl survey for Hake. So there's certainly a number of other species of krill that occur in the California current, and actually that's one of my questions that I have for this data is, we have to kind of lump these two species together because the frequency response is such that we can't discriminate them based on just the acoustics. If we did have net samples, we could have maybe perhaps seen Thysanowessus spinifera located closer to shore and more shelf waters, and Euphausia pacifica offshore. Right now, it's sort of just all lumped together, but I think it would be really exciting. We've been talking on my team about digging deeper into the species-specific differences, because that might explain some of the sort of the noise around some of our GAM results, for example, and their relationship to shelf waters and bottom depths and stuff. So yes, when we have a method net that we're going to be using. It's just a matter of balancing time on the survey. As many of you know, I'm sure, have been on the survey. There's only 24 hours in a day, and it seems like a lot, but when you're out on a boat and you're doing CTDs and tows and acoustics and processing fish, it's really hard to find the time to do extra samples, but we're working on it.
Great. So there's another question about, are there any plans to connect this work with larval or juvenile Hake distribution data, especially to inform the recruitment dynamic in future work?
Yes, I don't know who asked that question, but that is a huge focus of our work. Yeah, so right now the Hake, so we have the different age classes of Hake, and we can look at different distributions of Hake based on age, but the young of the year and the age 1 Hake are a really big focus because obviously that, could have a huge influence on their recruitment. And particularly tying in the ADCP data and looking at transport of these larval fish is an active area that we are trying to get going right now, actually. Yeah, so stay tuned.
Great. That's the last question that I have in chat. Beth, do you have any last things you want to say to us before we wrap everything up?
No, I'm just really thankful for everyone and all your great questions. And again, if you want to reach out, I'm happy to keep talking about Hake or krill or anything related to the California current. So just send me an email. And yeah, thanks again.
Wonderful. And if you are not sure how to connect with Beth, you can connect with me and I'll figure out how to get you the information that you need. Beth, I'll just tell you that right now you're getting all the virtual applause coming into the chat. Great work. Thank you so much. Very interesting. You don't get to hear all those things, but I just let you know that they're all coming in. So right, all the great things. All right.
Well, thank you very much.
All right. For everybody online, I hope to see you again next week, either at seminar or Science on Tap. And for Beth, thank you so much for spending time with us. It's much appreciated. As I said before, it's just great that you are sharing your time with us and sharing this information and all your hard work. So thank you very much. All right, everybody.
Thank you.
Meeting. Thank you.